Hi guys, it's the educator. Welcome back to my channel. And in this video, I'm starting a new series where I'm going to be dropping lessons topic by topic. And then I'm starting with histology of the gastrointestinal tract, the tubular part. So I've divided it into two parts: the tubular part and then the accessory organs part. All right, so are you a medical student in your second year or third year, and then you are offering histology? Then this video is definitely for you. If you are watching this video and you are not yet a subscriber to this channel, do also hit the subscription button so that subsequent videos I'm going to be dropping on histology, you are not going to miss them. All right, so the objective of this session, the first thing is we are going to identify the four basic layers of the gastrointestinal wall, and then we're going to describe the regional modification of the mucosa and also the muscularis, the muscle layer, and then the mucosa. Not to worry in case you are not too familiar with these terms. As we progress in the lesson, we're going to explain the steps. Then number three, we are going to distinguish between the segments of the small intestine. All right. Then fourth is understand the enteric nervous system. All right. Now let's move on. All right, for the introduction, diagrammatical representation of the gastrointestinal tract, right? And then what we are just looking at, or our concentration is the tubular structure. Okay, as you can see in this diagram, there's no liver, there's no pancreas, all those accessory organs are not here. It's just the tubular structure. So the tubular structure starts from the esophagus down to the stomach, and it goes to the duodenum. Okay, from the duodenum, it goes to the second part of the intestine, and then to the third part of the intestine, and then it continues to the large intestine, and from the large intestine it goes on to the rectum, and then down to the anus. Okay, so for the introduction, the digestive system is responsible for the injection, for the digestion of dietary substance, and also the absorption of nutrients and the elimination of waste products. Alright, the secretions of the associated glandular organs such as the salivary glands, the pancreas, liver, and the gallbladder aid the GI tract in accomplishing these functions. Alright, that we are going to treat in a separate video. Okay, the accessory organs, we're going to treat that in a separate video. This video, it is just the tubular structure we're dealing with. The general plan in the gastrointestinal tract. Now we have that we have already acknowledged that the gastrointestinal tract is a muscular tube, and then this muscular tube is lined by mucous membrane with a consistent structural plan throughout. And the consistent structural plan includes number one, you have the mucosa. Mucosa is what lines the GIT mucosa, and then after mucosa, we have the submucosa. It is very easy to remember. So for mucosa, then what is under the mucosa? You have the submucosa, right? But now this mucosa is made up of three components. It's very important for you to know the three components. The three components include you have your epithelium, okay, then you have your lamina propria, then you have your muscularis mucosa. So this muscularis mucosa, what it means is the muscle in the mucosa layer, because we have the muscle outside the mucosa layer. So that is why in some textbook you see it as muscularis interna and muscularis externa. When you see muscularis interna, it means that it's inside the mucosa. Then when you see muscularis externa, it is outside the mucosa. And muscularis externa is the proper muscular layer. Okay, so for the submucosa is made up of thick connective tissue. Remember, the lamina propria is thin, and then the submucosa is made up of thick connective tissue. It contains the infrastructure, right? So we have infrastructure in the body, right? We have arteries, we have veins, we have lymphatics, nerves. These guys are known as infrastructure. So this diagram here tells us the layer. So you have mucosa. Mucosa is made up of your epithelium, lamina propria, and then muscularis mucosa. Then you have your submucosa. Then you have your muscularis propria. Your muscularis propria is also known as muscularis externa that I talked about. And then you have your adventitia, which is the outermost layer. Okay, so now let's move on. Still under the general plan, the third one is the muscularis externa. It's regarded as the engine because it does the work of peristalsis. It is the one putting the work to move your food from one region to another. Okay, now this muscularis externa has a particular arrangement. Okay, the function of this inner circular layer is that it constricts the tube. Okay, while the outer longitudinal layer shortens the tube. So the two rows of inner circular and outer longitudinal brings about peristalsis. Okay, now moving on, we have the fourth layer, which is adventitia. So the adventitia or the serosa. Now you see it in your text. You see sometimes you write adventitia, sometimes you write serosa. While both of them are the last layer in the general plan of the GIT. You should take note that both of them are not the same thing. Okay, so don't use adventitia when you're supposed to use serosa, and don't use serosa when you're supposed to use adventitia. They are two different things. Okay, so when do we use adventitia? Alright, so for adventitia is a connective tissue with vessel and fat. Alright, with vessel and fat. While serosa is only in the peritoneal cavity. Okay, so when a particular organ is covered by peritoneum, it is regarded as serosa. It's regarded as serosa. Why, if it is not covered by the peritoneum, it is regarded as adventitia. All right. So moving on, we have the mucosa, which is what we are going to discuss first. The mucosa is made up of epithelium, it's made up of lamina propria, and it's made up of muscularis mucosa. Right. So for the mucosa, we'll be looking at four mucosas which you can see in the GIT. Okay, so the first mucosa we'll see is the protective mucosa. Protective mucosa. So it's known as protective mucosa because it carries out the function of protection, and you will see it in locations where protections are required. Epithelium, you can see from here down to this place. This is stratified epithelium. As you can see it's in layers. It's in layers. It's in layers, and it is strong. Okay, so where are we going to find this protective mucosa? We'll find protective mucosa in the oral cavity, in the pharynx, in the esophagus, and in the anal canal. So the next mucosa we'll be looking at is the secretory mucosa. The secretory mucosa. So the secretory mucosa, the structure, it is made up of specialized cells for enzyme and acid production. Okay, so where are we going to find secretory mucosa? We find them exclusively in the stomach. Okay, so when you hear secretory, we find them in the stomach. Okay, it's made up of simple columnar epithelium. Please take note of that. It's made up of simple columnar epithelium. And so the role is chemical breakdown of food. Absorptive mucosa. The absorptive mucosa, the structure of the absorptive mucosa, the features, firstly, we'll see villi, finger-like structures. Okay, so this is a villi. You can see it is a finger-like structure. You can see it looks like fingers. Looks like fingers. Okay, those are villi, and then we'll see crepes. Crepes. So these crepes are invagination to maximize surface area. So as we move on in the lesson, all these terms will be explained, but then just follow along. So the location, this is entirely found in the small intestine. So absorptive mucosa is found in the small intestine. So we can even use the name of the mucosa to know where it is located. Secretory, we know that it's stomach that secretes, so we know it is found in the stomach. Then absorptive, we know that small intestine absorbs, so we know that absorptive is going to be located in the small intestine. Then the role is primary site for nutrient uptake into the blood. Okay, so then the absorptive or protective mucosa. So we have the type of mucosa that carries out the function of absorbing and also the protective function. Okay, so how is the structure? The structure it is dense with goblet cell and straight tubular gland, no villi. So if you look at this, you see here, you see a lot of goblet cell. You look at this gland, you see another goblet cell. You look at glands, you see a lot of goblet cells. Okay, once you see a lot of goblet cells in a GIT slide, okay, one thing that should come to your mind is that is an absorptive and protective mucosa which is found in the large intestine. Okay, you know the large intestine carries the function of water absorption, okay, and heavy mucus secretion for waste lubrication. Submucosa is made up of tissues, like I said, and the tissue are dense, irregular connective tissue. Okay, so in this submucosa, there is a plexus. Remember, we said something like enteric nervous system in our objective. Okay, one of the objective was for us to understand the enteric nervous system plexus. That is number 4. All right, so that is what we are currently doing. Submucosal plexus is known as the maintenance plexus. The maintenance plexus. So what it does is to regulate glandular secretion. Plexus does. Now moving on, we have the muscularis externa. Remember, we said the muscularis externa has arrangement. We said that the inner circular layer squeezes the tube, all right, and the outer longitudinal layer shortens the tube. And as the tube is getting shortened, it is propulsing the food to the next region. Okay. Now moving on, for the muscularis externa, the plexus that is found there is the myenteric plexus. Myenteric plexus. So you can see it is something that shouldn't be forgotten. You have submucosa, and then submucosa is submucosa plexus, right? And then you have muscularis externa. Muscularis externa is myenteric plexus. Now, this myenteric plexus is known as the alpha plexus. Okay, so the location, it is sandwiched between the two muscular layers. So if this is a muscular layer, for example, and then we have this to be the inner circular layer, for example, this is the inner circular layer, and then this is the outer longitudinal layer. Then the myenteric plexus, they are here. This is where they are found between the inner circular and outer longitudinal. So what is their function? Their function is for motility. You know, muscles generally they carry out the function of movement, motility. So you know, submucosa is related to secretion. Then the movement is controlled by the myenteric plexus. We are going to look at the individual structures right away. So the first one we're looking at is the esophagus. All right, the esophagus. That's starting from the beginning. the esophagus. So the primary function of the esophagus is for rapid transportation of food from the pharynx to the stomach. So the esophagus connects the pharynx to the stomach. Okay. Now remember, we already said that the mucosa in esophagus is a protective mucosa, right? The epithelium is non-keratinized stratified squamous epithelium. So please take note of this: non-keratinized stratified epithelium. Now you can see a question to tell you, why is the esophagus non-keratinized stratified squamous epithelium, or what is the benefit, all right, or what is the functional histology of esophagus having a non-keratinized stratified squamous epithelium? It provides maximum protection against mechanical abrasion of raw food. I said that earlier, raw food like dry gari or crusty bread. Okay, so moving on, we have the lamina propria. The lamina propria is relatively sparse, contains mucous glands near the stomach, which is the cardiac glands, to protect against acid reflux. Okay, so the submucosa, which is the next after the mucosa, contains esophageal glands. Okay, so now looking at this histological slide that you can see here. Okay, let me show you what I am talking about. So if you look at this region here, so this region here, you have your mucosa. This region here, you have your mucosa. Then if you look closely from here down to this place, you have your lamina propria. Your lamina propria. Okay, so this is your epithelium, which I circled, and then you have this arrow here to be your lamina propria. So everything here represents the mucosa. Everything here represents the mucosa. And then if you move on from here to here, represent the next layer. And what is the next layer? The next layer is the submucosa. So this is the submucosa. Now the feature of submucosa, it says it contains esophageal glands, right? So if you look closely, you see something here. This is a gland. If you look closely again, you see that this is a gland. All right. If you look closely, you see this is a gland. Those glands are known as esophageal glands. All right. So moving on to the outer layer, we have adventitia mostly. Adventitia mostly. Since the esophagus is in the thorax and attached to the surrounding structures, it lacks a serosa until it pierces the diaphragm. Before you can discuss any GIT structure, you start with mucosa, right? From mucosa, you go to submucosa. From submucosa to muscularis proper. From muscularis proper, you now go to the adventitia or the serosa. So anything you want to discuss, if it is esophagus, if it is stomach, small intestine, large intestine, you must follow this pattern. Okay. So now this is the slide of esophagus. How do we know this is the slide of esophagus? Now, if you look at this, remember this is the epithelium, right? Epithelium. This is the epithelium here, and then we have these guys here to be the lamina propria. Lamina propria. Then these guys here, from here like this, and it goes like this, just like that, round, round. Then like this. This makes up the submucosa. Okay, this makes up the submucosa. So this one's here are the submucosa. So if you look closely, the submucosa, you'll be seeing some glands. Those are the esophageal glands. If you look at this guy here, this is a gland. This is a gland. Then if you look at the structure here, this is another gland. Okay, let's see if we can see any other gland. Okay, I'm not seeing any gland again. All right, so moving on, we now have the muscularis externa. Okay, the muscularis externa. Now, if you look at the muscularis externa, you see that it's a bit larger than the other layer somehow. That is because of how the muscularis externa is structured. Okay, so we have two layers here. Two layers. If you look at it here, we have the inner and then we have the outer. We have the slide of esophagus, but then we are concentrating on the muscular aspects, the muscularis externa. So there's something peculiar about the muscular aspect of the esophagus, or the muscularis externa of the esophagus. And this is known as the rule of threes, the rule of threes or the rule of thirds. Okay, so what exactly is this rule of thirds? Now, the muscularis externa of esophagus is unique because it's transitioned from voluntary to involuntary control. So the upper one third of the esophagus is made up of skeletal muscle only. Alright, it's made of skeletal muscle only, which means that the initial stage of swallowing is voluntary. So the reason why you can intentionally swallow is because the muscular layer in your esophagus, one third, is skeletal muscle. So middle one third, like I said, the middle one third is mixed. Alright, it is mixed. Okay, you have a combination of skeletal and smooth muscle fibers, which means that there's a point where you swallow, you get to the middle of your esophagus, you can control the food at that level. Okay, you can control the food at that level depending on which muscle is dominant at that point. If it is the skeletal muscle, you can control it. If it is the smooth muscle, of course, involuntarily, so the food just goes down like that. Then the lower part, which is the one third, the lower one third is made up of smooth muscle only, which means that once food gets to that point, which is smooth muscle only, there's no way you can control that food again. All right. So for the applied histology, okay, this transition is why disorders like achalasia, failure of the smooth muscle to relax, or gastroesophageal reflux disease, GERD, primarily affects the lower part of the esophagus. All right. So now we are moving on to the next, which is the stomach. All right. So the stomach is a very, very important structure in the GIT, and then not only being that it is important, it also has a lot of subdivisions. Okay, and we are going to be explaining the subdivision. So we know that the stomach is a J-shaped structure. Okay, so we have the stomach to be like this. Okay, we have the cardiac parts, then we have the fundus, then we have the body, and then we have pyloric part. Okay, so this is the cardiac C, then this is the fundus F. Okay, and then this is the body, and then this is the pyloric part. So now that is why I said it is very important for you to know your growths. Now, if you look at this structure, this is the coordination, and esophagus is just here like this. So let me say this is esophagus. So what part of the stomach directly meets with the esophagus? It is the cardiac part, right? So if you look at this slide projected here, you see that this slide is a mixture of two structures in the GIT, and that brings me to the different junction in the GIT. Okay, so if you look at this, this is the partition here. Now, this whole thing you can see here, this is the esophagus. If you look at this, you see this is stratified squamous. Okay, so what I'm seeing here, this is stratified squamous, and then if you look at this here, see that it is an entirely different stuff. It means that we have transited to another structure. Okay, so from the esophagus then to the stomach. So this is the stomach. To be more precise, this is the cardiac part of the stomach. Because after the esophagus, you have the cardiac part. Right? So this is the cardiac part of the stomach. But then I'm still going to tell you why it's the cardiac part of the stomach. So let's just run through the introduction of the stomach. So the stomach is an expandable organ. Okay, it is designed to store large meals and mechanically break them down. It is made up of rugae, which are large longitudinal folds of the mucosa and submucosa. So when it's mucosa and submucosa folds forms the rugae, the function allows the stomach to distend, and as it's distending, it does not tear. Okay, so this rugae, the folds, allows it to extend. You already know the secretory mucosa. We've talked about the secretory mucosa already, so we're not going to waste so much time. So moving to the muscularis externa. The stomach has a different orientation when it comes to the muscularis externa. Remember, we have inner and the outer layer for the normal muscularis layer. But when it comes to the stomach, it's totally different. So we have inner oblique, we have middle circular, and we have outer longitudinal. You know, why is the stomach designed, or why is the stomach structured like that? It's structured because this third layer allows for turning and lingering motion to turn food to chyme. Okay, to turn food to chyme, which is going to be moved to the duodenum. Alright, so this is the expanded version. So now, why is this the cardiac part of the stomach? This is the cardiac part of the stomach. How do you know this is the stomach first of all? When you see pit, when you see pit, you should know the GIT epithelium is simple columnar. It is just that the beginning, which is the esophagus, is not simple columnar. The beginning is stratified squamous, right? Stratified squamous, non-keratinized stratified squamous. So what we're going to see here is pit. So you can see structures, you can see pits like this pit. They are known as gastric pits, right? Gastric pits, and this gastric pits connect to gland. So we have something like this in histology. We have something like this. We have something like this. So this here is the pit, right? This is the gastric pit, and all of these guys you can see here, they are the gastric glands. Gastric glands. Okay, the gastric gland. So now, what is the feature that tells us that this is the cardiac part of the stomach? It is when the pit and the gland have almost the same size. They are equal. They are equal. So let's look for a gland since we have seen a pit. So if you go down here, you see, okay, this is a pit. This is another pit, right? So if you look at it, this is a gland. This is a gland. Then you have another gland. So by the time you look at it, you see the pit and the gland have equal size. So that gives you a clue that that is the cardiac portion of the stomach. Another clue is most of the time they intentionally give you a slide showing the esophagus. So when they give you a slide showing the esophagus, there's no need of you to think twice. You should know that what is next after the esophagus is the cardiac part of the stomach. Alright, so moving on, this is another part. Looking at this, this is a histological slide of the fundus and the body of the stomach. right? So the fundus and the body of the stomach both have similar histological slides. So how will you know that this is the fundus of the body of the stomach? All right. So what you should take note of is that the pit. Whenever you see a pit, you should know that you are looking at the histological slide or histological section of the stomach, gastric stomach, related to stomach, gastric pit. Now, if you look at it, the gastric pit is shorter than the glands. Look at one gland, look at another gland. You can see how long the glands are. You can see how long the glands are, and look at the pit. It's short. So once you see something like this, you should know that okay, this is the fundus and the body of the stomach. You should know the cardiac. Okay, for the cardiac, you have the pit and the glands are almost equal size, if not equal. While for the fundus and the body, which is represented here, you have the pit to be shorter than the glands. So the glands here are known as fundic. The glands here are known as cardiac gland, and the glands here are known as fundic gland from fundus. All right. So moving on, this is also histological section of this slide here is the stomach. So let me say this is slide A and this is slide B. Both slides are histological slides of the stomach. But then how do we differentiate which section it is? Okay, so if you look at it, remember I said the first criteria is your pit. Alright, so look at the pit. Look at the pit. This is the pit. Alright, this is the pit. Okay, this is the pit. And then if you look at this, you see glands. To the point that glands are reaching this point, you see glands from here are reaching this point. So if you look at the length, the pit is short, the glands are long. So this gives us a clue that this is fundus or the body of the stomach. Now, if you look at this, you see that the pit here is longer compared to the glands. So what am I trying to say? If you look at this, this is a gland, this is a gland, this is a gland. But now, if you look at it, look at the pit. Look at how deep the pit is. This is where the pit reaches. Oh, you should take note. This is where the pit reaches. You can see the length of the pit. This is also, we can also use this. This is pit. This is where the pit reaches. So the gastric pit is longer than the gland here. So this represents the pylorus part of the stomach. So we can regard these glands that you are seeing here as pyloric gland. Alright, so moving on, the gastric glands in the stomach. This is another important concept that you must take note of while you are doing histology of the stomach. Okay, so the gastric pit and the glands. The mucosa is indented by deep gastric pits. I've shown you that already, which leads into tubular gastric gland. Alright, so we can see here that this is the pit. This is the pit here. And now this pit is giving rise to tubular glands. Tubular glands. Alright, so now in all of these, there are cells, important cells, key cells that you must take note of. You can't do gastric glands without knowing all of these cells. So the first one is the parietal cell, which is known as the ozontic cell. They are large, triangular, and they are fried egg in appearance. Okay, so once you see any cell that has this fried egg appearance, you know that is the parietal or the ozontic cell. So what they secrete HCl and intrinsic factor, which is essential for vitamin B12 absorption. Okay, so moving on, the next one is the chief cells. Chief cells. Okay, just a mnemonic. Chief always take Pepsi. So which means that the chief cell, which are known as the zymogenic cells, they secrete pepsinogen, which is inactive, right? So the pepsinogen, that is an inactive enzyme, HCl now converts it to pepsin to digest protein, and this HCl is found in the stomach. So this means that without the parietal cells secreting HCl, these chief cells, what they are secreting cannot be used. So they will just keep on secreting pepsinogen. If it's not converted to pepsin, then protein is not going to be digested. Then we have mucous neck cells. Mucous neck cells are found in the upper part of the gland. Okay, so this is a gland. So this is a gland here. This is a gland here. Okay, this is the pit. This is the neck of the gland. So you look at it, these are the mucous neck cells. Okay, so the endocrine cells, also known as the G cells, they secrete hormones like gastrin directly into the blood to stimulate acid production. Okay, so moving on, this is a junction. This is the second junction I'm looking at. So remember, the first junction we looked at was the junction between the esophagus and the stomach, and that junction is known as gastroesophageal junction. Gastroesophageal junction. The next junction we're looking at here is known as the gastroduodenal junction. Gastroduodenal junction. This tells us that there are two slides here. Remember I said it's a slide of gastroduodenal, right? Gastroduodenal. Gastroduodenal, which means that there's stomach and there's also duodenum. So if you look at this from here down like this represents the stomach. This represents the stomach, right? Then let me use another color so that you can understand what I'm talking about. Then from those points down like this represents the duodenum. Alright, so now getting to the duodenum, how do we know that we are looking at the slide of the duodenum? One important feature of the duodenum is the Brunner's gland. So if you look at this closely, you see that there are some glands here. Okay, in higher magnification, you will appreciate them very well. Alright, so you see that there are some glands. These are the glands here. These glands are known as Brunner's gland. Okay, so moving on, we have the small intestine. So basically the small intestine mission is to maximize contact between the nutrients and the absorptive cells, right? So there are three levels of food in the small intestine. You have the plica circularis, which is known as the valves of Keckering. Okay, so they are large, they are permanent circular folds of the mucosa and submucosa. They slow down chyme and increase area by 3x. So in case you are wondering what the 600x rule means, it means that Plica circularis increases area by 3x, villi increases area by 10x, microvilli increases area by 20x. So if you multiply everything together, you get the 600x. Okay, so villi are basically finger-like projections of the mucosa. Only, please take notes. Plica circularis is mucosa and submucosa. Villi is mucosa only. And in this mucosa, they contain what is known as the lacteal. Lacteal is lymph vessel for fat absorption. Okay, that can still form part of your MCQ. Right, then we have microvilli, brush border. So microscopic folds on the apical surface of individual enterocytes. It increases the area by 20%. So the total surface area increases. Combine this, make the small intestine roughly the size of a tennis court. I know you guys can agree with me on that. Alright, so moving on, this is the intestine, comparison of the intestine. So if you look at this, this is a Brunner's gland. How do I know that this is a Brunner's gland? It's a Brunner's gland because of this. So you have this gland here, this gland here, this gland. So you can see there's no pit per se. If this were to be a stomach, we call it gastric pit, but we can't really say this is a gastric pit because we're not seeing villi, right? So say villi are finger-like projection. So this is one finger-like projection. This is another finger-like. Then this is another finger like that. This is another finger like that. See, they look like fingers. So when you see something that look like finger, know that you're already in the intestine. So you should forget about stomach. Okay, so this is representation of the duodenum. Alright, so most importantly, we should not forget that there is a general plan. Right, we talked about general plan in the beginning of the video. We said that we have the mucosa, submucosa, the muscularis proper, and then the adventitia. Okay, so if you look at this, it might be a little bit confusing to say which one is mucosa, which one is submucosa, which one is adventitia. You should know that your Brunner's gland is in the submucosa. So we already know that this guy here is the submucosa. So which means that from here above, we now have the mucosa, right? We have the mucosa. And then if you know the mucosa is made up of the epithelium lamina propria and then the muscularis mucosa. So if you look at this one I'm circling here, you can see it's a muscle. It's a smooth muscle. You might not really appreciate it well, but then this is a smooth muscle. So this is a mucosa, muscularis mucosa. Now if you look at this is the epithelium here. So this line I'm drawing here, this is the epithelium. And this guy inside here, this is the connective tissue, which is the lamina propria. The same thing is applicable to this guy over here, the slide B. So this is slide B, and then this is slide A. So remember the unique difference for duodenum is the Brunner's gland. The unique difference for ileum is lymphoid aggregates. Lymphoid aggregates. So whenever you see lymphoid tissue, lymphoid aggregate, lymphoid follicles, you should know what you're looking at is the ileum part of the small intestine. Anything relating to lymphoid, you should know it is the ileum. Alright, so now if you look at this, it is still the same general plan. Okay, you see the same general plan. You have this guy here to be the villi. Okay, this is your villi. This is also your villi. Okay, we said finger-like structures. This is also your villi. But now you see that in this particular slide, there are a lot of, if you look at this is the submucosa, right? This is the submucosa. Have you seen any gland? So we're not seeing any gland, but then we have seen a villi. So this tells us that it is a small, it is an intestine, small intestine, right? But then we don't know the exact intestine. So we check the submucosa. Is there gland? There's no gland. So we're left with ileum and then jejunum, which is this side, right? So ileum, you will see lymphoid tissues, you see lymphoid aggregates. So if you look at this slide very well, you see that there are a lot of small purple stained substances that makes up the lymphoid of the ileum. All right. So this slide here is the slide of the ileum. Okay, slide of the ileum. All right. So moving on, this is just the lacteal, the lacteal that is present in the mucosa villi of small intestine. So moving on, we have the duodenum in proper. The duodenum is made up of Brunner's gland, right? We said that already. So what exactly is the function of this Brunner's gland? Remember I said it is very important for you to connect your function with histology. That's why we have functional histology, okay? So why do we have Brunner's gland? We have Brunner's gland for a reason, right? So the duodenum receives chyme. Chyme is acidic because it's from the stomach. A lot of enzyme has acted on it together with HCl, acid. So as that chyme is coming to the duodenum, something needs to neutralize it so that the duodenum is not going to have ulcer. So the acid is not going to burn the duodenum. So which means that there is a need for a gland, right? So that's why we have a gland in duodenum. That gland is known as the Brunner's gland. So these are massive pale staining glands located in the submucosa. All right, located in the submucosa. So this is our submucosa. This is our submucosa. So this is one Brunner's gland. This is another Brunner's gland. Okay, so the function, they secrete a rich alkaline mucus, say that neutralizes the acid that's coming. Okay, now for the jejunum, how do we now know jejunum? All right, so jejunum have tallest villi. So when you see that the villi is tall, very tall, your mind should be coming to jejunum, but that's not the only reason to know that it's jejunum. Okay, another thing is that they have prominent placa secularis. Prominent placa secularis. All right, so the pious patches of ileum is found in which part of the intestine? It's found in the ileum. So if you check, if you don't see Brunner's gland, if you don't see pious patches, then you know that it is jejunum. No need to think too much. Then we have the ileum. The ileum have shorter villi. Ileum have shorter villi and more goblet cell. So if you see any intestine, small intestine, that has goblet cell, what should come to your mind is that that thing is ileum, right? That's a lot of goblet cell, but then it's small intestine, it's not large intestine. What should come to your mind is ileum. The function of pious patches. It is part of the gut. The gut is known as the gut associated lymphoid tissue, right? So basically what they do is to prevent infection before they reach the colon, which is the large intestine. Before they reach the large intestine, pious patches act there. Alright, so moving on, we have the large intestine. The large intestine, presence of Krebs, and also the water recovery system. Alright, so the mission basically is to absorb water, nutrients, and formation of feces, and also the lubrication of feces. Okay, so in the large intestine, there's absence of villi. So if you look at this large intestine, this you will see that there's no villi. This thing here is not a villi. Okay, you will see that there is no villi. The cells population in large intestine. What cells are you going to find in large intestine? You are going to find the enterocytes. Okay, and the work of the enterocyte basically is for water and salt uptake. You're also going to find goblet cell. In fact, when you hear large intestine and they ask you for cell population, the first cell that should come to mind is the goblet cell. And then you might want to ask why the goblet cell. Okay, it is because as water is removed, feces becomes dry and abrasive. Massive amounts of mucus are needed for lubrication. Where will this mucus come from? If there is no mucus, the feces is not going to be lubricated. And if you are passing stool, it is going to be a tug of war. So because of that, there is need for this lubrication, which is done by mucus secreted by what? The goblet cells. Okay, so that's another population of cells that you will see in the large intestine. Okay, so let's see if we can still appreciate the structures in this slide. So we have already identified our mucosa, right? Once you see the mucosa there, it is not looking like a villi, and it has goblet cells. So many goblet cells, which come to your mind is large intestine. There's no two ways about it. It is definitely a large intestine. Okay. Now, if you look closely, you have your um submucosa around here. You see your submucosa, and then your laminar propri. Okay, let's go to the next slide to appreciate it better. Okay, so we have something like this here. So you can see this. You have your mucosa right here. I know mucosa is epithelium, laminar propri, and muscularis mucosa. Okay, so if you look at this guy here, you see that this is looking like a muscle, right? It's looking like a muscle. What kind of muscle? This is the muscularis mucosa. It is within the mucosa. You can still see the glands here, and then you can see goblet cells, goblet cells, goblet cells. So this tells you that this is the mucosa. Okay, and then you will now see that after the mucosa, what you have the submucosa, right? So these guys here, all of these guys here, you can see makes up bulk of the submucosa. Okay, then as you proceed further, you will now see what you see your muscle. You see your muscle layer. Okay, you see your muscle layer. So this is your muscle layer, muscle layer, muscle layer, muscle layer, muscle layer, muscle layer. All right. So modification of the external muscle layer. Now, what are the modification? The inner circular layer is complete, but the outer longitudinal layer is unique. It is thickened into three distinct longitudinal bands called tenia coli. So you appreciate tenia coli more in the gross anatomy of the large intestine. This band acts like an elastic wristband, pulling the colon into sac-like pouches called the haustral. So now for the serosa and adventitia, what do we have? Do we have serosa in the large intestine, or we have adventitia, or we have both? Okay, so we have mostly serosa, which means that the large intestine is mostly intraperitoneal, which means that it is inside the peritoneum. Most of the large intestine are inside the peritoneum. Okay, so another thing you should take note of is that there is no Paneth cells. There is no Paneth cells. Okay, Paneth cells are found in the small intestine. Okay, but then they are absent in large intestine. All right, so moving on, this is a slide of an extension, or we can regard it as an extension or part of the large intestine. So this is the appendix. This is the appendix. Okay, so I just decided to include this slide. For some schools, they might not necessarily concentrate on the histology of appendix, but then just take note of this. All right, so you can see a lot of um, lymphoid tissues. Okay, see a lot of lymphoid tissues. You see it. Once you see it, and then it's looking like this, don't forget your muscular layer. Okay, you can see your muscular layer here. You can see your muscular layer here. Don't forget that this is your mucosa. All right, so mucosa. If you zoom in, you'll see that this is the epithelium. Okay, this is your epithelium here. All right, so moving on, we have the junction. At this now, you will see that you have two epithelial lining. Okay, if you look at this, you see this is looking like your stratified squamous. Okay, if you increase the magnification, then if you look at this one here, you see simple columnar. All right, you see simple columnar, which means that from simple columnar transiting back to squamous, it is telling me that I'm heading towards the anus. All right, the anus is also stratified epithelium. Remember, we said it in the beginning of the lesson that these places need protection, and so because of the protection it needs, the reason why it has the stratified squamous epithelium. So we have simple columnar epithelium here. This is the simple columnar epithelium, and then we have the stratified. So I'm giving you a clue that this stratified squamous epithelium here is the anus. So in the comment section, drop the answer. Let me know if you truly understand this lesson. Which junction is this? So there are two junctions that will come to mind: ileocecal junction and recto-inner junction. So tell me, are they the same? If they are the same, indicate in the comment section. If they are different, then pick one. All right, so moving on, we have the inner canal. The inner canal. Okay, so the pectinate, also known as the dentate line. So this is just a little bit of gross. Okay, this is the most important landmark. It represents the site where the simple columnar epithelium of the rectum abruptly changes. All right, so the three-step transition. The first one is the upper part, that is above. This is the rectum. This is the um pectinate line. This is the pectinate line here. Your pectinate line. Okay, it's your pectinate line here. and this guy here above is your rectum. Okay, this is your rectum here. And now above the upper part, that's above it, you have the simple columnar epithelium, similar to the rectum. Similar to the rectum. So automatically I've answered the question in the previous slide. Right now, I've answered the question. Okay, I've answered it. So this is the rectum here. Above it, you have simple columnar. So this is going to be simple columnar. The middle part is stratified columnar. So this part here, this line here, this pectinal line is stratified columnar or stratified cuboidal. Right, then the lower part is non-keratinized stratified. So this is non-keratinized stratified squamous epithelium. So we have gotten the answer already. So if you go back to this slide, we have simple columnar, right? Transiting to squamous, transiting to stratified squamous, non-keratinized. There's no keratin here. Okay, so this means that if we look at this place, this middle here, we can see stratified cuboidal or stratified columnar. So this tells us that this is the rectoanal junction. Rectoanal junction. But still indicate in the comment section. Let me know if you got the question. Alright, so inner columns. Okay, this is still more of your growth. Alright, so moving on, we have the inner canal, the musculature, and the support. So we have the internal inner sphincter and the external inner sphincter. You'll be hearing a lot of this in your growth very well, but then it is important that you know the histology. What is making up the histology? Okay, so the internal inner sphincter is the thickening of the inner circular smooth layer, and it is controlled by involuntary nervous system. Involuntary nervous system or autonomic nervous system. The external inner sphincter is composed of skeletal muscle from the pelvic floor. So because it's skeletal muscle, it's definitely controlled by voluntary. If it is smooth muscle, it's involuntary. So these are basic physiology that you should know. Alright, then the submucosa contains rich hemorrhoidal venous plexus. So enlargement of these veins leads to hemorrhoids, which is known as pile, commonly known as pile. Alright. So moving on, this is a summary of everything we have done in this lesson. So if at all you still find histology of the GIT, the tubular parts, difficult, then this table is definitely for you. Alright, what you need to do is to practice a lot of slides. Histology can be fun. The reason why people shy away from histology or run away from histology is the slide. Once you get the slides, once you know that okay, this slide is esophagus. Why is the esophagus? This slide is anus. Why is the anus? This slide is simple columnar. Why is it simple columnar? Okay, so for esophagus, we have the epithelium is stratified squamous. Once you see stratified squamous, and they tell you GIT organ or GIT structure. Now, oh, stratified squamous. What's stratified squamous in GIT? Anus and esophagus, right? That those are the two things that should come to your mind. Then the distinctive feature is skeletal and smooth muscle mix. Remember, we have the rules of thirds or the rules of threes, right? The first layer of the muscle is skeletal. Second part, that's the second one, third is combination of smooth and skeletal. And the third part is smooth only. Then we have the stomach. Stomach is simple columnar. The stomach, duodenum, jejunum, ileum, large intestine, everything is simple columnar. That's why we say that the epithelium of the GI tract is mainly simple columnar. So we can say the epithelium of the GI tract is simple columnar, but that is going to be wrong because esophagus is part of the GI tract. The inner canal is also part of the GI tract. So because of that, we can't fully say that it is made up of simple columnar only because of the stratified squamous of esophagus and the inner canal. Alright, so this table gives you an overview, a summary of distinctive features of each epithelium, or rather each structure. So what are you expected to see in a particular structure? So that okay, this is that. So for your stomach, you see gastric pit, right? Remember we said it. Once you see gastric pit, once you see pit, that pit, just know you are looking at the stomach. Okay, so at any point you go for an examination, you don't know the exact portion of the stomach, if it's cardiac or fundus or the body or the pylorus. Just put your stomach, at least you get a mark for that. But it's quite important that you know how to identify the parts. Alright, duodenum, Brunner's gland. Once you see gland in the submucosa, please take note of the location. It is in the submucosa, not anywhere. Alright, it is in the submucosa. For the jejunum, we have simple columnar also, where you see tall villi, tall villi, tall villi. The villi are tall. Alright, then the ileum, you see pious patches, which are gut associated lymphoid tissues. Alright, so long intestine, large intestine rather, there's no villi, but what you see there are many goblet cells. Alright, then the inner canal is stratified squamous, and then there's a transition at the pectinate line. Alright, so with this, we'll come to the end of this lesson under the gastrointestinal tract, the histology of the gastrointestinal tract. I know you enjoyed this video. Please drop a comment in the comment section. and most importantly, ensure that you subscribe to this channel. So if this is the first time you're watching my video, please do well to hit the subscription button. That is how I get encouraged to make more videos. Okay, and most importantly, please indicate in the comment section which video you want me to make next. All right, so I made this video based on demand, and so I'm going to be making the next histology video also based on demand. So just indicate in the comment section what you want me to make next. All right, and most importantly, share this video to your friends, share this video to your classmates, share this video to anybody you know who is a medical student or anybody who is offering histology. Bye for now, guys.